In this second video, we're going to take a look at what we can do to get even more energy out of our glucose. And we saw in the first part that the carbons in the glucose were oxidized to form CO2. And this energy was used to recharge for ATP molecules. And I ended the last video by discussing how there's still a lot over energy, or a lot of leftover energy that's stored on our electron carriers, NADH and FADH2 that we can see in this figure here. And you can think of these electrons as like little balls. If you put them on a table, they're just going to start bouncing because they've got so much energy that is stored up inside of them. And how can you get energy out of those little bouncing balls is what cells have spent millions of years through evolutionary processes trying to strip that energy out of there and use it. Because remember, the more energy a cell is going to have available, the more it's going to be able to do, because this energy is used by the cells to do work, and that work includes reproduction that is out there. So how do we get the leftover energy that is on these electron carriers transferred into usable batteries for the cell to use, our ATP molecules? And that's where we come to something that's really quite a novel thing known as an electron transport chain that's used by our cells that are out there. And if we look at this process here, an electron transport chain is going to use these high energy electrons. These energy-rich electrons are going to get dropped off with carriers that are built into a membrane. So either the plasma membrane for a prokaryotic cell or that inner mitochondrial membrane that folds back and forth for our eukaryotic cells. And these high energy electrons are going to get dropped off onto a series of molecules that are built into the membrane there, and as they're transferred from one molecule to the next molecule, a little bit of that energy is coming out of there. So you can almost think of it as these electrons are like balls that are bouncing, and we drop them down some stairs. And the first stair, it bounces a whole bunch because there's still a lot of energy there, but a little bit of that energy is going to get transferred out. And in this case, that energy is going to be used by this membrane protein to actually start pumping protons, or H plus ions, which are hydrogen with a positive charge, across that membrane. And if you think back to our discussions about membranes, charged molecules cannot easily slip through membranes. So these protons are effectively being trapped on one side of the membrane. And when you do this process here, we're going to build up more and more of these protons on one side of the membrane as our electron energy is being transferred to basically drive these pumps here. The other product that comes out of this is at the end of these series of transfers, you have to have somewhere for these electrons to go. And at the very end of this process, these electrons are going to be dumped onto an oxygen molecule. And this is the whole reason why we are breathing in. We're breathing in oxygen that's going to be used as what's known as an electron acceptor. And the oxygen is going to get dumped, or the electron is going to get dumped onto this oxygen. And now your oxygen has this negative charge associated with it. So we want to shield that negative charge with a positive charge. And there's where our hydrogen ions come into place. And effectively, what this produces is water. So the oxygen that you're breathing in right now is actually not being used to make carbon dioxide. It's being used to make water molecules. And if we look at this process within our cells that are out there, those protons that have built up over time, they're going to be potential energy. And so what I like to think of here is those protons are like water behind a dam. Here, we're pumping protons across a membrane, and if you look at a dam that's used to generate power, think of all the water that is evaporating and it's going up into the clouds, and then it rains and it runs down and it gets stored up in that lake behind the dam. Well, that's the equivalent here. That is potential energy when that water falls through the turbines in the dam. For our cells that are out there, in this case our mitochondria, those protons are being stored up in a lake on one side of the membrane, which is the equivalent to a dam. And just like with hydroelectric power, when you let water flow through the turbine in the dam, the turbine spins and that's being used to generate electrical power. Well, here, the turbine is a molecule that is known as ATP synthase. Again, this is buried in our membranes, and when the protons flow through our ATP synthase, it's going to start spinning like a turbine in the dam, and that energy that is coming out of this flow of the protons is actually going to be used to generate our ATP molecules. So effectively, the electron transport chain is generating our proton gradient over here, this potential energy that's stored behind our dam, which is our membrane. And then when our turbine spins and that proton flows through there, that's going to be used to recharge the batteries for the cell. And this is kind of a hard process to visualize. And so I found of this video that we can use to see this process. And so if we look at this, our high energy electrons are getting dropped off by our electron carriers, and those are being used to go through this series of electron transfers with the molecules in the membrane. And effectively, as those electrons are bouncing from one molecule to another in our electron transport chain, the leftover energy that is coming out of this is going to be used to pump our protons across the membrane. We build up this gradient here, and that is going to flow back through the membrane again to restore the balance of our proton concentration, and that is going to provide the energy for our ATP synthesis. And again, over here you can see there is your oxygen molecule where your electrons are ending up at the end of our electron transport chain. And if you think about this process, if we take the oxygen out of here, then you're going to run into some problems. Your electrons are going to back up on your electron transport chain, which means your protons are not being pumped across your membrane, and without your proton gradient, your ATP synthesis is going to stop. So effectively, in the lack of oxygen, a lot of cells that are out there aren't going to be able to replenish their batteries, and those cells are going to run into some problems. They're going to die. And if we go back and take a look at our little mitochondria within the cell here, we can put all of these parts together. Glycolysis is going to oxidize some of those carbons in the glucose molecule when you split the sugar, and that's going to result in 2 ATPs being produced, and also some of those electrons are going to go to form NADH that's going to be saved for later. We take the pyruvic acids, transport them into the mitochondria, where they're going to run through the citric acid cycle, and that's going to generate 2 more ATPs. So if you look at this process, by oxidizing our carbons in the glucose, we're actually able to get 4 ATPs net out of this whole process. But the leftover energy that was in that glucose molecule is now being stored up in all of these electron carriers that are floating around inside of the cell and the mitochondria now. They're going to go into our electron transport chain, and the energy from that is going to be used to pump the protons across the membrane. We generate our water using our oxygen as our electron acceptor at the end of that process, and that proton gradient is going to provide enough energy for the synthesis of about 34 ATP molecules by that enzyme ATP synthase. So if you look at the overall process here, we're generating a maximum of about 38 ATPs per glucose that is being oxidized inside of the cells here. So if we look at what's going on with this process here, if we take the mitochondria out of this equation and you think about it, glycolysis is just going to result in the production of 2 ATP molecules from the oxidation of glucose. So not really very many batteries are being made for the cell to use. This process does require the presence of oxygen as our terminal electron acceptor for your electron transport chain to function effectively here. And this leads to the question, well, what does a cell do when there's no oxygen around? And what the cell can do is effectively switch their electron transport chain. At the very end of that process, they use a slightly different molecule that is going to take those electrons that have given up their energy for the electron transport chain, and you're going to put those electrons onto an alternative electron acceptor. Cells can use things like nitrate molecules, Or they can use carbonate or sulfate molecules and do basically the same reaction. We generate water, they're going to generate other kinds of compounds as a result of this process that's known as anaerobic cellular respiration. The problem for the cells is that when they do this process here, they're going to make fewer ATP molecules. They're not going to get up to that maximum amount of around 38 ATPs per glucose. Then we have the last situation. Well, what does the cell do if it has no electron acceptor available for their electron transport chain? There's no oxygen. There's none of these other inorganic molecules floating around. And when that happens, what the cells are going to do is switch over and use the process that's known as fermentation here. And when we look at this process of fermentation, what the cells are doing with fermentation is they're effectively using an organic molecule as an electron acceptor. They still run through glycolysis. They still invest 2 ATPs and they generate 4 ATP molecules and get that net 2 ATPs coming out of this process, but then they've generated this NADH molecule as a result of glycolysis. And if they don't get more of this oxidized version of their NADH, known as NAD+, then glycolysis is going to shut down. And if glycolysis shuts down, those cells are going to have no energy available, and they're more than likely going to die as a result of that. So by using fermentation, they can effectively take that NADH and dump those high energy electrons that are stored on there. Rather than electron transport chain, they put them onto an organic molecule and they regenerate their oxidized version of their electron carrier that can go back into glycolysis. The end product, when you look at fermentation, depends on the kind of cells that you're looking at. An example that we are familiar with is when you look at yeast that you can find in beer or wine or even in bread production. Those yeast are taking the sugar, and in the absence of oxygen, they can run it through glycolysis, and then they ferment the leftover products and they generate ethanol and a little bit of CO2. And this is what gives us our carbonation when we look at a lot of beer that is out there, but also this is what causes your bread to rise. The yeast bubbles of carbon dioxide that are coming off are what are causing our bread to rise. For our muscles, or if we look at bacteria that are inside of milk that are used for the production of cheese, they're going to be fermenting sugars again, but here they're going to generate a molecule called lactic acid. So overall, when we look at this process, fermentation is really being used so that glycolysis can continue, because we need these electron acceptors available for that process to work, and we get a little bit of ATP out of there. If we look at the overall process here and we look at our catabolic reactions, remember we're eating. We are constantly ingesting food that is out there. And your food contains multiple types of organic molecules. We looked at this catabolic reactions for glucose. So you run it through glycolysis and all these other side reactions here. And if we look at this, what we can see is your food contains other organic molecules. It's got fats, it's got proteins, and it's got nucleic acids that don't appear on this diagram. All of these molecules here that are available in our food are going to get catabolized. They're going to get broken down into smaller and smaller pieces. And if you look at this, fats, when we look at triglycerides, they're made of glycerol and fatty acids. And those carbons that are in there are storing up energy, just like our carbons in sugar. When we catabolize or break down these carbon bonds in these molecules, we will actually take them and drop some of these intermediates into various parts of these series of reactions. So glycolysis can use some of the molecules from fats in terms of glycerol. We can also take some of our fatty acid molecules that are out there and break 2 carbons off at a time, and that gives us those acetyl groups that are going to drop into our citric acid cycle. And both of these sets of reactions are still going to generate our ATP molecules, and they're going to generate those electron carriers that can be used in electron transport chain, where we get a whole bunch of energy out of this process. Likewise, when we look at proteins, you break those peptide bonds to release your amino acids, and those amino acids, depending on which type, can drop into various different sets of reactions here to generate our ATP and also for use in our electron transport chain.